My name is Joan, by the way. I'm the director of the staff's facility and associate professor in the forensic science department. I approached the dean of the College of Criminal Justice about a year after I arrived here in 2006. So about 2007, I approached him about the possibility of developing a facility of human decomposition to do taphonomic studies and then ultimately have anthropology, a collection. And the Board of Regents, Tricia Pollard, she was an advocate for it, and so we had some internal funding and we started construction in 2008. The original mission of staffs was to make it a multidisciplinary facility where students and researchers and law enforcement officers that needed training could come and collaborate on research or training.
My name is Sheree Hughes-Stam and I'm an assistant professor of forensic science here in the department. I have three main projects at the moment. The first is federally funded by the National Institute of Justice, and it's looking at improved tissue preservation for mass disasters. So the NIJ project is looking at preserving human tissues. We are concentrating on skin and muscle samples and preserving them at room temperature. So this is important for our response after a mass disaster, because often the electricity is lost and there is no way to preserve the tissues from all of the victims of the mass disaster. So we may be looking at hundreds or thousands of bodies that are decomposing very quickly. And as that's occurring, the DNA in those tissues is degrading very rapidly, which makes identification of those victims extremely difficult. So what we're looking at is devising a simple method for preserving those skin and muscle tissues without refrigeration, so that we can then quickly identify those remains later on. The second project is looking at improved DNA typing from incinerated teeth. So this is improving the way we can genotype and understanding how we can get better DNA profiles from bodies that have been incinerated, so bushfires, house fires, etc. So Samantha, the student on that project, was at staffs and removed the mandible and the maxilla from 3 cadavers. And we divided them in half and did half before burning and after burning. We were looking at the teeth, both histologically and what the structure of the teeth are, and then after burning, the DNA identification as well. So what we wanted to identify was, were there particular teeth that were more vulnerable to damage, so therefore we can identify the teeth that would be more reliable for DNA identification after incineration, and also particular regions of the tooth. We wanted to know whether the regions of the tooth, whether it was the crown or the root of the tooth, that was more preserved, so we could get better quality DNA from those particular regions.
My name is Samantha Tiffin and I'm in the Master of Science in Forensic Science program here at SAM. During my first year in this program, I worked as Dr. Hughes-Stam's graduate assistant. And so when she came over from Australia, I got to meet her, understand her interests, specifically merging the fields of anthropology and DNA. And so we bounced a lot of ideas between us, and we finally came across the Incinerated Human Teeth project. So in the past, when there's been a mass disaster such as an arson or a plane crash that involves fire, bones and teeth are the last resort for DNA. So what I'm looking at is focusing on two different aspects of the teeth. One is looking at the cellular structure of the teeth to determine if there's something going on within the tooth that can affect the success of a DNA profile. And second is trying to find a correlation between the cellular structure of the teeth and the success of my DNA profiling.
And the third project is looking at preserving forensically relevant insects that are associated with the decomposition process.
My name is Sybil Buchelai. I'm an assistant professor of biology at Sam Houston State University.
And I'm Erin Lynn. I'm also an assistant professor in biology at Sam Houston State University. So the project that we're working on really involves looking at what bacteria are present on a human cadaver and how that might change over time through the process of decomposition. But also, how is that relevant to the decomposition process in terms of gas production and subsequent insect recruitment?
We place the cadavers out at specific time points, and then through very controlled experimental design, we visit the staff's facility regularly to collect the specimens. We're collecting bacteria and insects and some gas samples as well at these specific time intervals. I like to think it's a very charismatic research project, and we get a lot of, we grab their attention with it. A lot of them are interested in biology, but forensics as well, and so we have several graduate students on the project. Some of them are more interested in the microbial side, some of them more interested in the entomology side, and then we have probably a dozen undergraduate research assistants that assist with our graduate researchers. And so the graduate students are, they're generating their master's thesis from this research project. And then the undergraduates, some of them gain research experience and then they go on to different jobs. And then some of them transition into the graduate student role from these projects.
And all of the students, I think, are involved in every aspect of the project, especially the graduate students. Our graduate students we have are involved in experimental design, logistical planning, sample collection, data analysis, and they publish and they present at local and regional and national meetings. So student involvement is really high, and they're involved in pretty much all aspects of this research project.
What staffs can offer to the academic community is those students that are interested in chemistry but have sort of a forensic liking or lean towards forensics, or in biology, they have the ability to do research that you're not going to be able to do all over the world. There's only 6 of these facilities in the United States, and most of the world are not permitted to do research on human remains. So in that sense, they have this really unique opportunity to do research. Also, for the criminal justice system, we're able to work with law enforcement to actually using bodies to say, yes, this body's been dead this long, based on the research we're doing here. If you use human analogs such as pigs or rabbits, they're not the same. And so this is a very unique opportunity.